Okay, so I'm gonna help you visualize what actually happens in meiosis. Okay, so here we have a cell and we have the chromosomes already condensed, so you can expect this one is actually in prophase. Any prophase happens like this, but in meiosis it starts with prophase 1. Okay, so in prophase 1 you have um, the process of crossing over. Before crossing over can happen, you must pair the homologous chromosomes together. Okay, now it's easy to know whether a chromosome is homologous to one another by generally just seeing the size. So this one is the same size, so usually the same size is homologous chromosomes. Okay, so what's going to happen in prophase 1 is one part of the maternal chromosome is going to overlap with the paternal chromosome like this one, and these two are going to snap a piece of that part, okay, and then put it on the paternal chromosome, and that piece from paternal chromosome is going to attach to this part. So I'm not going to do it here, but you know how it goes. Okay, now that process, crossing over, it's not compulsory. It can happen or not happen at all. Okay, so let's assume in this case that process does not happen at all. Okay, so in prophase 1, so remember the nuclear membrane disintegrates and the centriole will go to this part of the cell, opposite poles, one on this one, one on this one. Metaphase 1 is when the homologous chromosomes are aligned side by side. Okay, prophase 1 just now, actually it's still not aligned yet. Okay, so metaphase 1, so metaphase 1, the homologous chromosome aligned side by side, and then anaphase 1, okay, goes right there, goes right there, okay, and then telophase 1, this one is going to crinkle together, and then we have one cell on this side with one set of chromosome and another with one set. Okay, now imagine this is already one cell, so prophase 2, prophase 2, okay, there's not much really happening in prophase 2 because it's already replicated and whatever, it's already condensed. Let's just proceed with metaphase 2. So metaphase 2, remember chromosomes, sister chromatids are still attached with a centromere. So chromosomes align at the equatorial plane, and then the spindle fiber will attach to the centromere from both sides. So one chromosome, there's two spindle fiber attached to it. Okay, and then we proceed with where's anaphase? Where's the thing? Oh, there you go. So anaphase, these two sister chromatids will be pulled by the spindle fibers to one side. This one will also be pulled to one side, and then during telophase, okay, one cell will undergo cytokinesis, and then we have one cell with one set and another cell with one cell. Okay, and then at the end you have 4 cells that have one set of chromosome, and then you have it, meiosis complete. Okay, bonus video right here. Okay, now remember when I said in metaphase 2 chromosomes are arranged at random on the equatorial plane for each daughter cell. Now what does this actually mean? Okay, now I mentioned that this phase allows for variation in chromosomes. Variation is kepelbagaian. Okay, so I'm gonna show you what that means shortly. All right, now over here we have the arrangement of chromosomes in metaphase. Okay, now the thing about random assortment, arranging at random, it can happen at any metaphase. Metaphase in meiosis 1 or metaphase in meiosis 2. Okay, now over here we have the arrangement when you are in metaphase 1. So homologous chromosomes are aligned side by side at the equatorial plane. Okay, so you can see right there, there's the crossing over happening. So this one changes the piece with the other homologous chromosome, this one also down there. Okay, now what it means by random when it is arranged at random is that these two homologous chromosomes can be at either side. Okay, so this one can be here, this one can be here, or this one can be here. Okay, or this one can be here, or this one can be here, whatever. So what's gonna happen is, if the arrangement during metaphase is like this at random, any side, so one side goes here and one side goes here. Okay, so one cell, so if you divide the cell into two during metaphase 1, okay, so this cell is going to have this set, this one is going to have this set. Okay, both red, both paternal, both maternal. Okay, now sometimes you can arrange this one goes right here during metaphase. Okay, so this one stays the same right there. Oops. Okay, so during anaphase, this one goes to the other side, this one goes to the other side. So one cell has a different set. So because the chromosomes are arranged at random, okay, there is many ways. There are many ways the daughter cell can end up having what chromosome. So it can have two paternals or two maternals, or one maternal with the new combination of gene, or the one that doesn't. It depends. All right, so now in metaphase 2, the chromatids, the arrangement of chromatids in one chromosome can also be arranged at random. Okay, when I say random, you can arrange them like this, or you can arrange them like this. Okay, or you can arrange them like this on the equatorial plane, or you can arrange them like this. Okay, so as a result, when these two sister chromatids separate, okay, I'll show you one example. So I do it right. Okay, so when they separate, during anaphase 2, okay, they separate, go. Okay, so you have two new cells, two daughter cells, okay, that has this combination. Okay, now imagine if during the metaphase plate, the metaphase 2, so if during metaphase 2, okay, the arrangement is like this, the other way around. Okay, so if during metaphase it's like this, then during anaphase, the sister chromatids separate. Okay, so you have over here a new combination that has a different arrangement of chromosomes. Okay, so that's why when during metaphase, the random arrangement of chromosomes are really unique in making meiosis, producing variation in cells. Okay.